next lecture will be performed by Dr. Richard Mooney, who is first-time lecturer here. And I would like to welcome him very warmly. Richard Mooney is the George Braff Geller Professor of Neurobiology in the Department of Neurobiology in the Duke University School of Medicine. He is motivated by long-lasting interest for neuroscience and music. His major research interests are understanding how songbirds learn to sing and how mice produce and perceive the innate ultrasonic vocalizations. So for those of you who are worried about mice is missing on our conference, I think he will make it up to you. Professor Mooney, the floor is yours.
Great, nice to be here. Good afternoon, morning, evening, wherever you may be. I'm delighted to be here, and I'm going to give you a short talk on these two topics that are in my title, which is analyzing vocalizations and vocal circuits in mice. And I will move pretty quickly. A fair amount of this is published, but towards the end, I'll show a little bit of unpublished data. And just as Victoria was alluding to, my background is primarily in birdsong, but over the last few years, we've turned our attention to encompass vocalizations in mice. And just to be clear, we're talking about ultrasonic vocalizations typically produced by male mice during courtship. But what I'm going to say today, actually, as far as we can tell, also applies to female mice. I'm not going to show much or any data from females, but we've been able to duplicate most of what I'm talking about as far as brain circuitry in female mice. Now, just again, by way of background, I studied birdsong for a long time. I'm very interested in birdsong still, primarily because birds are remarkable vocal mimics. They can, at least songbirds can, as juveniles, memorize and then copy the song of an adult conspecific. That's a remarkable example of imitative learning, and we've mined that very, very heavily. But even in the highly derived circuitry of the bird, there's a realization that the ground structure for vocalization is that's highly conserved through probably all vertebrates, or at least all birds and mammals, and that is a brainstem circuitry that's obligatory for vocal gating. And I'm going to talk quite a bit about that in the circuit part. But first, a little bit about behavior. It's really completely intuitive with birdsong that if you're looking at a zebrafinch song sonogram like that shown on the bottom of this image here, that one could say, well, they're discrete elements and these are grouped together in the case of the zebrafinch in stereotyped sequences to make phrases. And those phrases are repeated in long bouts. There's a lot of structure that's both stereotyped at the local level as well as at a more global level. And we refer to these as songs. And it's understandable that people studying vocalizations in other animals, and particularly in animals that produce vocalizations during courtship, would refer to them, those vocalizations, also as songs. I would like to also point out if one just takes a snippet of ultrasonic vocalizations from a mouse, as shown at the top here, there looks to be a lot of structure. There is a lot of structure. Appears to be a lot of repeated structure. And this kind of close-in view bears a lot of similarities to what one sees when one looks at a sonogram of a songbird like the zebrafinch. And we were really curious, and we being my collaborators, John Pearson and Jack Goffinet, as well as a student working in my group, Sam Brudner, really how far we could take this, or how far we could push this analogy, or conversely, how far we could distinguish these two types of vocalizations. And to do that, Jack and John adapted a method. This is an unsupervised learning tool known as a variational autoencoder. It's a dimensionality reduction method originally applied for image analysis. And they had the insight that it could be applied to analyze sonograms. In this case, USV syllables shown on the left. And the autoencoder has two qualities, one of, well, or several, I should say, but two major steps, one of which is an encoder that takes the raw spectrogram, roughly 16,000 pixels, and compresses it into a relatively low dimensional representation. I mean, here shown as 64 dimensions, but in most applications, we use 32 dimensions. And in fact, almost all of the variance is described by 7 or 8 dimensions in the latent representation, this compressed encoded representation. And the autoencoder trains itself by coupling this encoder step with a decoder, which converts the latent representation back to an image. And when those images that are output are close matches to the input raw spectrograms, then learning is effectively complete. And that decoder ability is, in my opinion, quite powerful because it is a generative capacity of the algorithm that allows you to probe the latent space, measure distances in latent space, which is very handy for assessing similarity, but also interpolating between real embeddings to generate smooth transitions like this transect between two real mouse USVs generates this smooth transition in structure. So there are a lot of cool things about it. This is published this spring in eLife, and the software to run this is available from a site listed in that paper. We're happy to help anybody who wants to apply this to their system and help you use it to study things you're interested in. But what I want to turn to is just the contrasts that emerge when you use a tool like this to study mouse USVs versus bird songs. Here's a male zebrafinch in the upper left, and here's a male mouse in the middle. And below are UMAP projections from the latent embeddings of zebrafinch song, shown as purple clusters, and from mouse courtship ultrasonic vocalizations. These are really, really large, I would say very large data sets, typically many tens of thousands of individual renditions. And what one sees in the UMAP projections from the latent embeddings of bird song is that there are very tight clusters that if I were to decode these would correspond to individual syllables in the bird's repertoire. That is, there's a lot of space in between different syllables. There's not continuity. Now, just the opposite emerges when one takes a similarly large data set of mouse USV recordings. This is from an individual mouse, and projects them onto a two-dimensional plane, as shown here. There's no obvious clustering. Now, there are many tools for studying mouse USVs, and some that annotate what are termed distinct syllables, and there's a lot of attention to the taxonomy of mouse USVs. And I will admit I'm not an expert in this, but I'm just pointing out that when one uses this sort of technique to, that is a syllable-based identifier like Muppet, and projects those labeled syllables onto the autoencoder UMAP representation, there are colored regions, but what one can see from this is that there's really continuous distribution of sound. There isn't strong segregation between different elements. Now, that's not to say that these animals don't perceive the colored regions as distinct syllables. There may be perceptual ways of warping this more continuous representation, but at least using an equivalent approach to analyzing both birdsong and mouse song, we don't see this evidence of strong clustering. Now, we can recover traditional features. So if you label, for example, the frequency bandwidth or maximum frequency or duration in these UMAP projections, there's a very nice gradation across the projection. So this tool is capable of capturing sort of classical hand-picked features that are used to label USVs and other vocal sounds, but it just doesn't return clear evidence of clustering. Now, as I mentioned at the outset, with zebrafinch song, there's also this higher level order, and one can take a variant of the autoencoder method, which we call a shotgun variational autoencoder, and just sample random snippets, typically several tens of milliseconds long, from the sound stream. and feed that into an autoencoder, and it finds most energetically satisfying solution that in the case of zebrafinch song just returns in the UMAP projection a continuous sequence of sound, which is the stereotype sequence of syllables in the parent song phrase. And these ribbons that you get from finch song are quite beautiful and have a lot of information packed in them, because again, these are from many, many renditions, and the width of the ribbon reflects the amount of variation that occurs in that syllable over those many renditions. Now, there's another feature here, which I didn't stress. I've just talked about syllables here labeled A through F in the finch song. There are these regions here, which are labeled as introductory notes, and birdsong experts in the audience will appreciate that these are innate vocalizations. They're distinct from the syllables that the finch copies from a tutor. And these innate vocalizations are much more sort of continuous in the space they occupy in the projection. Now, if you do the same thing with mouse USVs and apply the shotgun variational autoencoder, and I'll just play this for you, and I hope you can hear it. This is slowed down 4 times and dropped an octave further, so it's audible. But the again, what emerges is a really strong contrast. There isn't really strong sequence structure that emerges from this kind of analysis. So there's not strong clustering, nor is there strong evidence of syntactic stereotypy in the vocal repertoire. Again, you know, these are early days. We're happy to help other people. Obviously, with the conference that's focused on ultrasonic vocalizations, people are working in mice or rats or other animals. This would be a very useful tool, and happy to help you apply it in your own labs. Now, I'm going to turn now to in the second part of my talk to vocal circuitry and just introduce a part of the brain. First, introduce a former postdoc, Catherine Cheetah, and a current student in my lab, Valerie Michael, who've been the major drivers of this work. Katie is now a professor at Cornell University in Ithaca in the US, and starting her own lab on mouse vocalizations, and would be an excellent choice for people who are thinking of studying further, who, for example, might be interested in a postdoc. I can strongly recommend her. She's an excellent scientist, as is Valerie. And Katie and Valerie had a kind of a different approach. I would say from many years of working in birdsong, the approach has been strongly top-down in the field, partly because there are forebrain structures that are so obvious and so essential to producing birdsong. To make a mammalian or a human analogy, it's very corticocentric. And you know, that contrasts with a classical approach to circuit breaking for behavior, where you might be inclined to work from the bottom up, from the effector through the motor neuron pool to the premotor neuron pools and so on. And what Katie, I think, had, which was really a really powerful insight, is that in the really extensive mammalian literature on vocalization, a part of the midbrain periaqueductal gray, or PAG, has emerged as an essential node, an obligatory gate for vocalization. And this just shows a cluster of labeled neurons. I'll talk a little bit more about how we label them, but a cluster of neurons in the caudolateral PAG that have a very important role in gating vocalizations. And what I mean by obligatory is that there's a clinical literature in humans, and then also a very extensive experimental literature in mammals, showing that bilateral lesions in this region of the PAG result in mutism. They render animals or humans unable to vocalize. And this is true not only for innate vocalizations, but in humans for speech. So this is a really critical component of the speech network. Now, again, this is, you know, building on a tremendous foundation of work from many other groups, and particularly Uwe Jürgens. but Jurgens recorded in the periaqueductal gray, stimulated, lesioned, etc., and found units that were vocalization related. They could be active at vocal onset, particularly. They typically were not activated by playback, suggesting more of a motoric role. And that seems kind of straightforward in a sense, but when we started to look at this, we were struck by the lack of progress into kind of a modern circuit analysis. And the problem stems from the fact that the periaqueductal gray does many, many, many things, and many, fundamental things like regulating defensive behaviors, processing pain, thermoregulation, sleep, cardiovascular tone, respiration, on and on. Vocalization is just one component of that. And that, for an experimentalist, presents a real big problem because the PAG is a heterogeneous structure, both in terms of cell types and in terms of function. And these, from what we can tell from electrophysiological recordings and functional experiments, these many different behaviors map onto neurons that are commingled with each other, and at least at the time that we started this project, might include neurons that serve multiple functions, not just a single function. And what Katie and Valerie did was first map the region of the PAG that was highly active in male mice as they produced USVs in response to either a live female or female urine, and build up a consensus map, which in this raw data from one animal here really is this caudolateral part of the PAG. Now, this is immediate early gene mapping. By itself, it's not particularly useful. It just confirms that there is a region of the PAG that's a hotspot for vocalization. But at that time, we were collaborating, and still are, quite actively with Fan Wong, a next door neighbor of mine here at Duke, who recently moved to MIT. But Katsu Sakurai, who was then a postdoc in Fan's lab, and Fan developed what I consider an incredibly innovative and creative approach to genetically tagging and accessing transiently active neurons. And the gist of it is a knock-in mouse that expresses the avian viral receptor known as TVA under the FOS promoter. So the FOS is a protein encoded by an immediate early gene that scales with calcium influx in neurons. So neurons that are highly active express a lot of FOS, and in these animals, they also express this viral receptor, TVA. Now, the really cool twist in this is that it's an avian receptor, so it's essentially invisible to mammalian signaling elements. But more importantly, in my opinion, is that it was engineered to have a PES sequence, this DS or destabilizing component, so that the TVA receptor only stays expressed in the neuron membrane, in the active neurons membrane, for a few hours, roughly paralleling the expression of FOS. And so what one can do is, once one has this mouse and one has a consensus map of where a hotspot is for FOS expression for the behavior of interest, is then go in a mouse that's been challenged to make the behavior a lot, to vocalize a lot in this case, and then use a pseudotype lentivirus to inject virus into that hotspot where the TVA receptor is expressed at high levels. And this engineered virus has the cognate ligand that binds to the TVA receptor, and that can be used to internalize all variety of things to label and manipulate these neurons and trace their connections, as I'll describe in the next few minutes. So this was a strategy that Katie and Valerie used, and this just shows a way of approaching it using the FOS TVA mouse and these cane lentivirus Cree injections, along with a standard AAV flex channel rhodopsin. So this technique will get channel rhodopsin specifically in PAG neurons that were highly active during vocalization. One can wait a few weeks, implant a ferrule and a fiber optic, and then illuminate with blue light. And the result of this is shown here. A couple of features of this: the vocalizations only are emitted as long as the blue light is on, and I'll contrast that with some other experiments a little bit later. The other is that the behavior is really specific. I mean, I'm not going to show it here for the sake of time, but if one were to stimulate in this manner in just a pan-neuronally in the PAG, one would elicit a whole range of behaviors in addition to vocalization, particularly escape, ballistic jumping behaviors and the like. So we don't elicit that with this technique. As I said, this structure in the PAG seems to be a gate more than a patterning element. For example, the specific pattern of stimulation didn't seem to have any effect on the structure of the emitted vocalizations. The vocalizations were emitted in a stereotyped manner, that is in a USV typical manner, regardless of whether we used really short pulses or long pulses and so on. Now, as I mentioned, if one just activates the PAG non-specifically with a pan-neuronal approach and stimulates in the PAG, one gets vocalization for sure, but one also gets a huge amount of movement. The animal is running and jumping and engaged in active escape behaviors. With this cane-based approach, this intersectional approach, we see really no evidence of extraneous movement. The animal vocalizes, it's pure vocalization. It doesn't really seem to interrupt much of its ongoing behavior to do that. Now, just again, in addition to the gain of function experiments, one can use loss of function methods, and here using the same approach, but to introduce the tetanus toxin light chain into these PAG-USV neurons. This abolishes vocalizations in male mice, and it doesn't seem to interfere with other aspects of courtship. So they still pursue the female investigator, mount her, and so on, but they don't vocalize. And I should add, this, I'm not showing this here, but this allows you to test the idea that the vocalizations actually do something. This was something that Katie and Valerie looked at, and in fact, in a 3-chamber assay, female preference assay, they were able to show that males that could vocalize a lot, in contrast to these males that were rendered unable to produce USVs, had an advantage. They were more attracted to the female. Just a little more circuit breaking, one can map the axonal projections of these PAG-USV neurons. They go to several targets, but one in particular that is of interest to us is the nucleus retroambiguus, which is a respiratory premotor pool, also thought to house components of the vocal pattern generating circuitry in mammals and in birds. And this just activating this pathway specifically, again, is able to trigger vocalizations. All right, just, again, in the interest of, how you can use these approaches to really analyze circuitry, what's quite attractive to me about studying vocalizations in the mouse is that the PAG is truly an obligatory node for vocalizations, for producing USVs. And so presumably the decision to vocalize depends on how those PAG-USV neurons integrate information. And in fact, the emerging picture that I'll just give you a sketch of now is one of, really tremendous, telescoping of connectivity to weigh different kinds of information that could inform whether or not the context is an appropriate one to vocalize in. And, in the big picture, vocalizations are costly. I mean, they're energetic, but they're also costly in terms of advertising. And if one is to vocalize in a context that, is inappropriate, there's risk involved. And advertising advertises not only to potential mates or conspecifics, but also to potential predators. So there's a lot of, weight placed on this decision. It's not something that comes for free. And the appropriate context in mice is when a suitable mate is nearby or conspecific one wants to affiliate with or advertise to is nearby. And of course, an inappropriate context is one where there's somebody out there who wants to eat you. Katie and Valerie, or I should say Valerie and Katie, then systematically map connections onto these PAG-USV neurons. Again, I'm not going to go into great detail here. This is published recently in eLife, and the upshot of it is the PAG is a site of massive convergence, but two regions stuck out to us as a kind of, again, a starting point to analyze the circuit, one of which was the preoptic area, which is generally implicated in reproductive and courtship behaviors, and where stimulation can elicit vocalization as well. The other is a region of the amygdala we call here the extended amygdala. It's adjacent to the central amygdala. And for a variety of technical reasons, we've focused on that region and have, with kind of the rationale being partly practical, we could, and second, that there's reasons to suspect that the valence of inputs from the amygdala might be distinct from those from the preoptic area. Now, the first thing that stands out about these inputs is that they're largely GABA releasing neurons, whether they're from the POA or from the amygdala. The neurons that are providing input into the PAG-USV complex are predominantly inhibitory cells. And that said, they have opposite effects on behavior. This is showing what happens when we optogenetically stimulate the preoptic area neurons that project into the PAG. One difference you'll note is that in contrast to the effects of optogenetic stimulation of the PAG-USV neurons directly, stimulating this pathway produces long trains of vocalizations that outlast the period of stimulation. So there's some additional process, reverberatory process, or prolonged activation cycle that occurs when this pathway is stimulated. And I could point you to a really nice paper from Lisa Stour's group that expands on this topic of persistence in the activation of this input. Now, those are GABAergic inputs that somehow release the behavior. This input from the amygdala doesn't elicit vocalizations in isolated mice, but if Valerie and Katie probed its actions during courtship, one could see suppression of vocalization. So the male's on top, he's vocalizing, light comes on, the vocalization stopped. But again, to me, a surprisingly specific manner. The animal doesn't freeze, he doesn't flee. He seems to continue on with the other aspects of courtship normally, but just disengages the vocal display. And as I mentioned, these are also GABAergic neurons, and it's easy to see how a GABAergic neuron could suppress vocalization if it directly inhibited the gating neurons in the PAG. And in fact, Valerie showed that was the case through a series of intersectional optogenetic circuit mapping experiments and brain slices, where she demonstrated that the output neurons, these PAG-USV neurons that are tagged with M-cherry, receive direct inhibitory input from these extended amygdala neurons. So recording in conditions where only monosynaptic currents were preserved, Valerie could find evidence of direct inhibition, which was blocked by GABA A receptor antagonists, like the one shown here. Now, the POA circuitry obviously has to be a little bit more baroque, because the input from the POA is also GABAergic, but in fact it releases vocalization. And what Valerie was able to map out was that these preoptic area axons directly synapse on VGAT positive cells, that is local inhibitory neurons in the PAG-USV complex. And those neurons in turn directly inhibit the PAG-USV output neurons. All right, this leads to a circuit that looks like this, where two inputs converge on the vocal gating complex in the periaqueductal gray, and one directly inhibits them, those from the extended amygdala, and the other disinhibits the PAG output neurons, which gate vocalizations. Now the question becomes, how do these components of the circuit actually balance their drive both to the PAG, and in turn, how their activity is balanced by their own inputs to regulate the decision to vocalize? And I'll just show you very quickly in the last minute some unpublished data, and some kind of curious features about the circuit that are emerging already. One of which is that the neurons that promote vocalization in the preoptic area send axons not only to the PAG, as I already showed you, but they also send axon collaterals into the part of the amygdala that directly suppresses vocalization. And I'm not sure how well this will come across on the screen, but in this dual intersectional experiment, the amygdalar neurons that project to the PAG are labeled in red, and the green collaterals are from preoptic area neurons that also project to the PAG. So they're projecting directly on top of these cells, and in fact are also transmitting signals that are relevant to vocalization. So particularly in fiber photometry experiments, if one images the axon collaterals of these POA PAG projecting neurons, one can see in these calcium signals from populations of axon terminals from the POA neurons that there are bursts of activity that are associated with vocalization. And when one aligns this to the bout onset for the USV train, there's an increase of activity well before the vocal bout starts, and then persists as the animal vocalizes. So vocalization related signal impinges on the vocal suppressing neurons in the amygdala. And in fact, just as the POA neurons that project to the PAG are inhibitory, so are those that project to the amygdala. And in fact, they're many of the same cells. And again, in brain slice experiments, Valerie's been able to show that these preoptic area neurons directly synapse on and inhibit the amygdala neurons that project to the PAG. Now, lastly, just to close out here, in fiber photometry experiments in the amygdala, what we're starting to see is evidence that encounters that would be likely to suppress vocalization, particularly attack, and this is imaging in the aggressor mouse, a resident challenged by a male intruder, or in response to odorants that are strongly threatening. This is fox urine component, the predator urine component that can induce freezing and flight in mice. One can see that these neurons are very strongly activated by these kinds of signals. And again, these are the sorts of signals that presumably are counterproductive for vocal production during courtship. So I'm going to close there. Just thanks to the organizers for the invitation. Mention the people who did the work. Sam, I alluded to earlier, Jack and John, collaborating on the variational autoencoder, Fan and Katsu particularly, and then Katie Cheetah and Valerie Michael, who've done the circuit breaking work. Thank you.
Thank you very much for your talk. Very interesting. We do have some questions, so I'm gonna allow people who wanted to ask them to speak. Fabrice, I think you had a question. Would you like to ask?
I had a question. Thanks for the talk, but it was at the very beginning of the talk. Wondering, when you reduce your spectrum to 64 data, the squares, I was wondering if it implies normalization in time and in frequency range or not.
Did you say normalization in time and frequency range?
Yeah.
Yeah, I believe so, but I'd have to check with Jack on that.
Okay. Thanks.
Yep.
Okay, so there's another question from Schwarzik. Would you be able to ask it yourself, or shall I? Okay, so I'm gonna just read it.
Mm-hmm.
Dr. Mooney, do you think that the PAG neurons for USVs lie close to those mediating fight? Or what may be the reason that certain stimulation elicit USVs, whereas other elicit fight?
Right. We think they're very closely located to each other and possibly intermingled. I mean, I was moving kind of quickly, but when we express channel rhodopsin paneuronally in the same region, in this caudal lateral region of the PAG, we elicit behaviors that look like defensive behaviors. These are optogenetic experiments conducted in isolated mice, but they engage in ballistic jumping, very, very active behaviors. So at least a component of what looks like defensive behavior gets elicited by stimulating neurons in the same region that contains the PAG-USV neurons. Yeah, and the decision as to which of these vocalizations to trigger, I think is, part of what you can see in some of the input analysis to the amygdala, for example. I mean, the POA is sending information about the drive to vocalize, but at the same time, the amygdala could receive information about extraneous cues that are vocal suppressing. And presumably the balance of those two sets of inputs, we think a direct excitatory input that's conveying potentially, threatening or aversive conditions is weighed against the drive to produce the vocalization, which is coming, approximately from the preoptic area, but, is also determined largely in mice by odorant information that's coming from regions that are upstream of the preoptic area.
Okay, thank you very much for your answer. And we have another question. Jeffrey, would you like to ask it yourself?
Sure. Can you hear me?
Yes.
A great talk. Thank you. Have you been able to look at some aversive vocalizations and compare those to some of the appetitive calls? Of course, mice have sonic aversive calls, but there's been some work that you can get some of the more aversive ultrasonic calls with very careful experimentation. I wonder if you've had a chance to look at that and think about that.
Right. That's a really good question, Jeffrey. And the short answer is that when we use tetanus light chain toxin to silence the PAG-USV neurons, painful stimuli still elicit audible vocalizations in the mice. So there are two interpretations. The first one, which I favor, is that they're actually separate output gating elements from the PAG that gate different types of vocalizations. The other is that the total drive on the circuit in aversive conditions or in response to noxious stimuli is much greater and can trigger vocalizations even when the USV promoting limb is suppressed. But I tend to favor the first idea that they're actually independent output channels, and that's something that Katie is actively engaged in studying now.
Great, thank you.
Just maybe one more thing about that, Jeffrey. I mean, you're probably more aware of this than, or more informed about this than I am, but there's some evidence that the retroambiguous may be organized topographically into domains that are USV promoting or audible vocalization promoting. So if that topography is preserved at the level of the caudolateral PAG, there might be a mechanism to, differentially access those types of vocalizations.
Yeah, that's a great idea. Thank you. Perfect.
Yeah.
Okay, Dr. Hammett, would you like to?
Thank you very much for the great talk. I got the question. So doing some kind of analog to the synthesizers, yes. But for example, so this PAG-USV are some kind of oscillator of the stimulation of the PAG-USV cells produce the same type of ultrasonic vocalization, and then there is another structure, yes, which modulates the ultrasound. So are those episodes the same, or you've got the whole range of the ultrasounds? Yes.
Yeah, I mean, I think the question, if I understand correctly, you're asking is there a patterning capacity for the PAG itself? That is there an oscillatory element to the PAG that could help influence the structure of vocal patterns. And the short answer is we don't know, but the longer answer is that the history of chemical stimulation, electrical stimulation, and now optogenetic stimulation all would suggest that whatever the PAG is doing, it's really more permissive for allowing vocalizations than really, participating in the patterning of them. As far as we can tell, we don't see evidence of, the structure of the stimulation having any influence on the vocal pattern. And in kind of a surprising way, really. I mean, what looks like happens is that the activity from the PAG is integrated with the ongoing respiratory rhythm. And that, for example, PAG stimulation doesn't reset the respiratory rhythm. And that ongoing respiratory rhythm then interacts with the permissive signal from the PAG to enable vocalizations to be emitted. It really looks just like a gate. I struggled with that term for a long time, reading Jürgen's papers. I said, well, what's a gate? But it really does look like it doesn't somehow insert itself into the pattern generating network. And that's a little bit curious because you would think these neurons in the PAG are excitatory. You would think that their activity, if it directly impinges on the pattern generating node and retroambiguous, would be able to contribute to that pattern, but we don't see evidence of that so far.
Thank you very much.